Let's move on to the structure of air. Okay, air is an appendageal structure. Okay, so the part of air which is visible above the skin, it's called air shaft, and the part of air which is below, which is within the skin, it's called air follicle. Okay, and the air follicle could be divided into upper segment and lower segment. This upper segment can be further divided into infundibulum and isthmus, and the lower segment can be further divided into suprabulbar area and the bulbar area. So they are divided into certain layers. Okay, but what is the line of demarcation between these segments? Okay, so here between the infundibulum and isthmus, you have the sebaceous gland, attachment of the sebaceous gland, and between the isthmus and suprabulbar area, it is erector pili. Okay. And suprabulbar is a small area which is seen about the bulb. So this bulbar area contains air matrix, and this is the area where the cells multiply and develop into an air. Okay. So if we see in this picture, this a part of air which is seen above the skin is called air shaft, and this part seen within the skin is called air follicle. Okay, and between the surface of the skin to the attachment of the sebaceous gland, this part is the infundibulum, and between the sebaceous gland and the attachment of the erector pili muscle, you see the isthmus. Okay, and below the isthmus you see the bulb, and this small area is called suprabulbar area. Okay, and below which you see the bulbar area. Okay, this is about the longitudinal section of the air. If you take the cross section of the air, like that is transverse section, you see the innermost layer is made up of medulla. Okay, it's called medulla. The next layer surrounding the medulla is the cortex, air cortex, and surrounding the air cortex you see a layer called cuticle. Okay, and surrounding the cuticle you see a sheath, it's called inner root sheath. Okay, and surrounding the inner root sheath you see another root sheath, it's called outer root sheath. So first one is the innermost layer is the medulla, next one surrounding it is the cortex. Okay, this layer is the cuticle, and this layer is the inner root sheath, and the outermost layer is the outer root sheath. Okay, this is about the cross section of the hair. Okay, so next we'll talk about the three types of air. Types of air. So three types of air include Lenuga air, Wheeler's air, and the terminal air. Lenuga is the fine air which is seen in the fetus and is shed in utero. Okay, so fine air seen in the fetus and it's shed in utero immediately after birth. And these are fine airs, they are seen everywhere, and these are non-medullated. Whereas the terminal airs are the thick hair which is seen in the scalp, eyebrows, eyelashes, beard area, mustache area, the groin, axilla. Okay, so you see a thick hair in these areas, so it's called terminal hair. It's a coarse hair, and you see in scalp, eyebrows, eyelashes, axilla, groin, and beard area and mustache. Okay, so this contains the medulla. It's modulated. Okay, this is about the types of air. Let's talk about the air cycle. And this phase varies between 2 to 6 months. Three phases: anagen, catagen, telogen. Okay, ACT, anagen, catagen, and telogen. Anagen is the phase of growth, and catagen is the phase of involution, and telogen is the phase of resting. And it is easier to remember. It's like to remember the duration. It's 2 to 6 years, 2 to 6 weeks, and 2 to 6 months. And as I said before, the length of the air depends on the duration of the anagen phase. Okay, and in anagen phase, there'll be about 90% of the air. Okay, 90% of the air will be in the anagen phase, and 1% will be in the catagen phase, and rest 9% will be in the telogen phase. And in telogen phase, there'll be 1% of the total telogen air will be shed every day. Okay, you should remember this. So suppose there are 10,000 airs, 1 lakh airs in the scale. 9% will be 9,000. Okay, in 9,000 there'll be 1% of the air which is shed in the telogen phase. So 1% of 9,000 will be 90. So there'll be around 90 airs will be shed every day. Okay, this is about air cycle. Easy to remember these phases. It's like ACT. It's ACT. Okay, act. Let's move on to the another structure. It's the glands. Okay, so 3 glands in skin which is embedded in the dermis. First thing is the sebaceous gland, and there are 2 other sweat glands. This eccrine gland, okay, which is opening directly into the surface of the skin, and there is another gland which opens along with the air follicle. Okay, this apocrine gland. And all these glands will have 2 parts. One is the secretory part or the gland, and there'll be a conducting part. Okay, so in eccrine glands, the secretory coil, and the duct will be eccrine duct. And apocrine also, there'll be apocrine gland, and the conducting part is apocrine duct. And there'll be sebaceous gland and sebaceous duct. Okay, so these are the 3 glands in the skin, and we'll see about them in detail. So what is the type of secretion in sebaceous gland? It's a holocrine secretion. Okay, holocrine means the whole cell dissolves and is shed. In eccrine glands, it's merocrine secretion. The cells in the glands will produce vacuoles which contain the sweat, and these vacuoles are released outside. Okay, it's called merocrine secretion. In apocrine glands, the apex of the cell is shed. Okay, so that's why it's called apocrine. Apex is shed, and its type of secretion is apocrine. And what are the functions of these glands? This sebaceous gland secretes sebum, okay, which helps in the lubrication. And this sebum is made up of triglycerides, wax esters, cholesterol esters, and squalene. So triglycerides are the most important component of the sebum, and they will be on 50 to 60%. Okay, and there will be lubrication. Okay, the eccrine glands produce sweat. This sweat plays an important role in the thermoregulation. It helps in maintaining the temperature of the body through the evaporation of water. Okay, so thermoregulation. The sweat which is produced in the eccrine gland is isotonic. So in the secretory coil, it is isotonic. Okay, when the sweat moves along this eccrine duct towards the skin surface under the influence of certain hormones like antidiuretic hormone and aldosterone, the sodium in the sweat is reabsorbed. Okay, so the eccrine duct, the sodium is reabsorbed, so the concentration of the sweat becomes hypotonic. So in the coil it's isotonic, and the duct it is hypotonic. And apocrine gland also produces sweat. Okay, and it also plays a minimal role in thermoregulation. And where are these situated? These sebaceous glands are situated everywhere except on palms and soles. Except on palms and soles, they are situated everywhere. Okay, and there are certain glands, modified sebaceous glands, like in eyelids. These are glands of Meibomian or glands of Zeiss. Okay, and it could be Montgomery tubercles. Okay, around the areola, and it could be Tyson's gland in the penis. Tyson's gland. These are modified sebaceous glands. Okay, these are modified. So let's see where are eccrine glands situated. Eccrine glands are situated everywhere except on anogenital region, like in clitoris and labia in females, and in males it is absent over the glans penis. Okay, and they are also absent in lips and external auditory canal. External auditory. So where are these apocrine glands situated? These apocrine glands are situated over the sides like axilla and axilla, nipple, areola, complex, and in the anogenital region. Okay, and how are they regulated? So sebaceous glands are under hormonal control as well as nervous control, and hormonal control is by testosterone, and it's innervated by sympathetic fibers. Okay, sympathetic fibers. And eccrine glands are controlled by the hormones like ADH, antidiuretic hormone, and aldosterone, and they're controlled by hypothalamus, the preoptic center, and they're innervated by sympathetic fibers, but the mediator is acetylcholine. Okay, generally the mediator for sympathetic fibers will be adrenaline or noradrenaline, whereas in case of sweat glands it is acetylcholine. It's otherwise called as cholinergic fibers. It is also under the influence of emotions and like gustatory control also. Okay, in emotions, like when you get nervousness or types of stress you get sweat, and when you eat spicy foods you also get sweat. Okay, and apocrine glands are regulated by sympathetic fibers which are mediated by adrenaline. Okay, adrenergic fibers. So this is about the glands. There are three glands: sebaceous glands, eccrine glands, and apocrine glands. Okay, let's move on to the structure of nail. So nail is made up of nail plate which rests on the nail bed. Okay, this nail plate rests on the nail blade which is seen underneath the nail plate, and this is held on to the nail plate, held on to the skin by the nail folds. This area which is in the proximal part of the nail is proximal nail fold, and the lateral areas are the lateral nail folds. There is a thin structure which holds the nail plate to the proximal nail fold. Okay, this structure is called the cuticle. So this is the proximal nail fold, and this is the nail plate, and this structure which holds the nail plate to the proximal nail fold is the cuticle. Okay, and if you see the section, another section, you will see this is a nail plate and it is resting on the nail blade which is seen above the distal phalanx. Okay, so where do these nail plate get synthesized? These nail plates get synthesized in the nail matrix. Okay, this nail plate gets synthesized in this part of area called nail matrix. Okay, so this nail matrix contains keratins and they proliferate keratinocytes, and they contain keratinocytes and they proliferate. Okay, the proximal part of the nail matrix synthesize the upper part of the nail plate. Okay, and whereas the distal part they synthesize the middle part and the lowermost part of the nail plate. Okay, the proximal part synthesize the dorsal part of the nail plate, whereas the distal part of the nail matrix, synthesize the middle part and the undersurface of the nail plate. Okay. Generally, nail matrix is not visible. Okay, it is seen beneath the nail fold, the proximal nail fold, but sometimes it can be seen extending onto the nail plate. Okay, this moon shaped structure or crescent shaped structure which is visible in the nail plate, okay, this is nail matrix. The visible part of the nail matrix nail plate is called the lunula. Okay, this is about the nail. And you should know, fingernail grows at the rate of 2 to 3.2 to 0.3 millimeter per day. Okay, and you should know that the nail growth, okay, then fingernails grow at a rate of 2 to 3 per month, and toenails grow at a rate of 1 millimeter per month. Okay, so this is about nail. So let's go to the last part of the lecture, cutaneous receptors. Okay, so there are four important cutaneous receptors. First is Merkel disc, which is seen in the basal layer of the epidermis, the Merkel cells. Second is the Meissner's corpuscles. This is situated in the dermal papilla. Okay. The third important is Ruffini corpuscle. Okay, which is seen in the deep dermis. And others are Pacinian corpuscle. This is also situated in the deep dermis. Okay. So let's see about these cutaneous receptors. Except for Merkel cell, all the three corpuscles are encapsulated. And Merkel cells are seen in the basal layer of epidermis. The Meissner's corpuscles are seen in the dermal papilla, and both Ruffini corpuscles and Pacinian corpuscles are seen in deep dermis. Okay, and Merkel cell, as we discussed earlier, they are slowly adapting. Okay, whereas Meissner's corpuscles are fast adapting. Ruffini corpuscles are slow adapting, whereas Pacinian corpuscles are fast adapting. So, which sensation is mediated by these receptors? Merkel cell is mediated by it mediates the touch sensation. Meissner's corpuscles mediate the fine touch. Okay, these are concentrated in the finger pads, so you'll be able to appreciate the fine texture. So, fine touch is mediated by Meissner's corpuscles. These are situated in the dermal papilla and they are concentrated in the finger pads. And Ruffini corpuscles mediate the touch and pressure, and Pacinian corpuscles mediate pressure and vibration. Okay, so this is the site of these cutaneous receptors is important. Merkel cells are seen in the basal layer of the epidermis. Meissner's corpuscles are seen in the dermal papilla. Ruffini corpuscle and Pacinian corpuscles are seen in the deep dermis. So these two receptors, Ruffini corpuscle and Pacinian corpuscle, are situated in the deep dermis. That is why you need some pressure to have the sensation. So these mediate the pressure sensation. Okay, and you need some fast adapting receptors for vibration, sensing vibration. So that is why Pacinian corpuscles, which are fast adaptation, mediates a vibration. And for appreciating fine touch also you need rapidly adapting receptors. That's why Meissner's corpuscles, which are fast adaptation, mediates the fine touch. Okay, this is about the cutaneous receptors.